Hi, everyone. So this is lesson 5, part 2. In the first part, we looked at the innate immune response, and today we're going to be taking a look at the adaptive immune response. Again, this is a very general overlook of what happens in this part of our immune system, and the real adaptive immune response is really quite complex, but this will give you a good general oversight of what actually goes on in this immune response. So the top of this page in Canvas, it describes what the adaptive immune response actually refers to. And really, the big difference between your innate and adaptive immune response is that your innate immune response is very general and broad. It's not specific. We talked about that last lesson. The adaptive immune response is specific to a specific pathogen. And really, the big key idea is that this specific response will also involve memory. So creating antibodies that will recognize that pathogen if it invades our bodies again. And this is part of the reason why vaccines are so important. And we'll be talking about vaccines in a later lesson as well. So when you flip over to your OneNote page, I've started you off with a couple of thought questions. The first one says, how might the innate immune response be unable to control an infection? So I want you to think about these things, like why would a pathogen be able to get past that first line of defense, that first immune response? Hopefully, one of the things that will come to mind is pathogen replication rate. Because if a pathogen is able to replicate quickly and in large amounts, some of those cells will be able to evade our immune cells. And so they can get into deeper parts of our body and then start to infect our cells. So that's one reason why a pathogen might be able to get past that first line of defense. The second, and this one might be a bit more difficult to think of, but we've actually talked about this idea in past lessons as well, is that some pathogens are able to hide out from our immune systems. So pathogens may be able to hide from our immune system. So in something like HIV, that virus actually infects our T cells. And so it hides out in the T cells and we're not able to initiate an immune response against it. So some pathogens have evolved to be able to infect certain immune cells, and that makes it more difficult to be able to stop. The next question here says, what happens when the innate immunity fails to clear an infection? So hopefully that should be straightforward. If the innate immunity can't clear out a pathogen, then that's when the adaptive immune response will kick in. Okay, so adaptive immune response begins. And that's what we'll be taking a look at in this lesson. What is that adaptive immune response? What does that involve? And obviously from last lesson, we know it involves B cells and T cells, but we'll look into more depth of what those cells actually do. And then your last question here is, what is the difference between innate and adaptive immunity? And we already discussed that when we looked here in your Canvas page, and we've outlined the difference there. The big reason, or the big difference, I should say, is that your innate immune response is non-specific, okay? Broad attack on pathogens. Whereas your adaptive immune response is specific to a specific pathogen, okay? And it does involve memory, and we'll look again at that in a bit more detail going forward. The next thing I'd like you to do in this lesson is watch this video. So this is part 2 of the crash course video. And again, it does go into a bit more detail than we need, but it does a really good job of animating what the cells look like and what they actually do. So watch that and follow along in your OneNote. And there's just a couple of quick questions here. So you'll see our B lymphocytes or our B cells, and we'll talk about the different types of them. Same with the T lymphocyte or T cells. And I just want you to answer these short questions in relation to what you learned in the video. After you've done that, we're going to take a closer look at what these B and T cells are actually doing. So you might remember from the last lesson, we talked about how B cells, their main function is really making antibodies. Okay, and so this is what your antibody right here looks like. And so they can either present it on the surface of their cells, or they can be released and the antibodies can flow throughout our body. And that's why that part's called the humoral immune response, because these antibodies travel through our humor or our body fluids like our blood. So here I put, recall that the innate immune system contains cells that detect potentially harmful antigens and then inform the adaptive immune response about the presence of these antigens. So we kind of left off here last lesson, and we talked about this idea of antigen presenting cells. So an antigen presenting cell can be another type of white blood cell. It can be like a macrophage, for instance. And so when it engulfs a pathogen, it's going to present some of those pathogenic antigens on the surface of its cells. And our T cells are going to be the ones that recognize it and really kick off that adaptive immune response. So it says when the pathogens are detected, these antigen-presenting cells will phagocytize, eat the pathogen, and digest it to form many different fragments of the antigen. Antigen fragments will then be transported to the surface of the cell, where they will serve as an indicator to other immune cells. And the key thing to remember here is that pathogens have more than one antigen. So when a pathogen is kind of digested by this phagocyte or whatever this antigen presenting cell might be, there's going to be multiple antigens, because remember that each one of these antibodies is going to be able to recognize a specific antigen. And so we want to present as many antigens as possible to help kick off that adaptive immune response quicker. Okay. And that's why it takes some time for the adaptive immune response to happen. It's not as automatic as the innate immune response. So let's take a look at these T cells. And in your OneNote, so we talked about the antigen presenting cells. So highlight here why this is a big and crucial step. And then we're going to look at the T cell response. And so I've given you some pictures here of those two T cells. We're only talking about these two here. There is a third, but I just want you to highlight here their role in the adaptive immune response. So T cells and B cells cannot see pathogens on their own. So like we just said, they need to recognize an antigen. So they have these cell surface receptors, and they will recognize antigens from our antigen presenting cells. So that is called antigen presentation. This can kick off your two types of T cells. The first one are killer T cells, and like it sounds, they do the killing. So it says killer T cells recognize these antigen presenting cells and kill the infected host cells. So this would be, for example, like one of our cells, it's presenting it, and our killer T cells say, okay, you've got pathogen in, let's kill you, because we know it's been infected. So you can see what that looks like here. It recognizes it, it releases some chemical enzymes into our host cell, and that causes the cell to undergo apoptosis or cell death. Okay, so that helps to get rid of those infected cells. The other big player are these helper T cells, and this really leads into the next response with the B cells, is that these helper T cells activate B cells and more killer T cells. So they send out these cytokines, which again are chemical signaling molecules, and that activates more B cells and more killer T cells. Okay, so that's a very general overview of what your T cells do. Then we get into the B cells, and this is the big part in relation to memory. So we've talked about this before, your B cell secreting antibodies. This is what an antibody actually looks like if you were to break it down in its structure. So you can see this is where you would have the binding of the antigen. That's where it's going to recognize. And this VDJ section right here that you see on this molecule right here, this is just showing that this is the part that can change for an antibody, and we have billions and billions of different antibodies in our body. So the big thing, you have to think of it as like each antibody is a key on a chain, but that key chain has a billion keys on it. So trying to figure out what key fits a pathogen can take some time. And so that's why your adaptive immune response might take a week or two before it actually starts to work. And so if a pathogen is able to infect your cells much more rapidly than your body's able to respond, then those are the kind of things that can lead to trouble. And so when we've talked about Ebola in the past, Ebola is able to infect and cause damage in our bodies much more quickly than our immune system is able to adapt to. Some of the strains of Ebola anyways. So If your body's not able to respond in time with that adaptive immune response, then that means bad things. Okay, so with your B cells, we have two main types of B cells. So B cell can become an effector B cell that releases antibodies to stop microbes, and we'll take a look at that in a second. Or they can become memory cells. So these memory cells will have a specific antibody, only one type of antibody. So it's like it's that key on the keychain, and it will recognize a specific pathogen or specific antigen to initiate a response. Okay, so this memory B cell are the key to our vaccinations, right? Creating these in our body is what helps protect us lifelong against whatever we're being vaccinated against. When we take a closer look and look at what antibodies are actually doing, they can work to stop infections in three ways. One of the most common ways is by neutralizing a pathogen. So you can see here we have a viral cell and a bacterial cell. And basically, it's just surrounding these pathogens to inactivate it. So that way it can't actually get inside our cells, and it kind of just stops it. And then what comes along is our phagocytic cells, and those are the ones that are going to engulf it, eat it, digest it, get rid of it. So that's one mechanism. The other way is agglutination. So you have these antibodies come in, and it basically, think of it as like sticky tape, where it causes a whole bunch of these pathogens to stick together, to clump together. Just think about agglutination when we did our blood typing lab. And so they clump together, and again, then it's going to be easier for our phagocytes to come around and engulf the cells and destroy them. The third way, and this relates back to one of the things we talked about in our last lesson, is the activation of the complement system. And so by activating this, you can see here is our antibodies here, activating complement molecules. That creates these holes inside the cell membrane of our cells, and that leads to cell lysis. Okay, sorry, I said our cells, but it's a foreign cell. So this would be like the cell membrane of a bacterial cell. And so it creates it punctures holes inside the cell membranes of those bacterial cells or whatever the pathogen may be. And that leads to cell lysis or apoptosis. Okay, so again, follow along and fill in here the B cell response and what these mechanisms of antibody responses are. And then the last thing I wanted to talk about is something you've probably felt when you've been sick, and that's this idea of clonal expansion. So I said, have you ever felt clonal expansion before? And clonal expansion literally means it's cloning your B and T cells. And so you get this big increase in your white blood cells. And so if you've ever been sick, had a sore throat, whatever it might be, you might feel swelling in your lymph nodes. And your doctor, if you've gone into your doctor will generally try to palpate those. And so you can see you have lymph nodes all around the body here. One of the easier areas to feel them in is in your neck. And so you can feel when you get an infection that your lymph nodes are swollen. And that's because your lymph nodes are creating more B and T cells. So clonal expansion happens in the lymph nodes, and it takes 10 to 12 days. So once that antigen or pathogen has been recognized, we're going to have a big increase in B and T cells, and that'll be sent out through the rest of your body there. Okay, so it says binding antigen causes B and T cells to replicate. That's what clonal expansion is. And this creates a pool of adaptive recognition specialists, so we can respond specifically to whatever that pathogen is. One of the things I mentioned earlier too was that we have a ton of different B cells and T cells. So it says each one of us makes B and T cells with different receptors in the order of 10 to the power of 7 due to recombination, so genetic differences. But the potential to make cells with novel or new receptors is estimated to be up to 10 to the 13 B cells and 10 to the 18 T cell receptors. So basically what that means is we almost have an infinite ability to recognize foreign molecules. The problem is, though, it just takes a long time to get that recognition. Okay, so I put in a little wrap up here, just summarizing the B and T cell response and what they do. B cells, antibodies. They can clump the pathogens and neutralize them, which helps to increase phagocytosis, and it can also enhance the function of our complement system. T cells, our killer T cells, kill infected host cells. The helper T cells help to initiate the response of other T cells and B cells. And so this is a nice little overview of what that looks like, starting with your macrophage becoming an antigen presenting cell and kicking off the T cell and B cell response. Now, if you're interested and you find the immune system interesting, this actually goes into more about the T cell response and what that final battle of fighting off the pathogen looks like. So if you want, as an extension, you can watch that video. Otherwise, finish your notes up here, and that's it for this lesson. As always, post questions in the Teams chat if you have any. Thanks.